Thank you for inviting me, and I'm going to take you back to the lecture of Dr. Yamada, and I'm going to talk about how the gene expression affects the way we look. And what is gene expression? Basically, the gene expression is a process that starts with gene information from the DNA that is transferred via the RNA to the cytoplasm and creates proteins. And from there, of course, all the traits are created. Now, the American Academy of Dermatology in 2017 found that individuals with more youthful skin, their skin behaved younger. In other words, they had the same genetic profile as the individuals that were much younger than them. And of course, I'm not saying anything new. You know that, you look younger because of your genes. But then what they looked into is that the younger looking older women had increased activity in genes that are associated with cell replication, with response to oxidative stress, and with protein metabolism. Now we're getting into a more interesting area because now we're talking about the biology and molecular biology and signaling and how this signaling interacts. And also they found that the mitochondrial structure and the metabolism of the mitochondria was different in the younger women. Again, now we're talking about something that's more than the sum of its parts. We're talking about the gestalt that Dr. Yamada introduced previously. A gestalt is more than the sum of its parts, and it's basically all of those interactions in the mitochondria. When you're talking about structure, you're talking about a gestalt, but it's more than the sum. The gestalt emerges, and it's something very different than all the parts that make it. Now, the rate of expression of a gene is based on signaling. It's based on the location in the nucleosome is very important, the histone composition, nucleosome occupancy and positioning within the chromatin. Chromatin is like an agent that takes all the parts of the genes and puts them back again together. It's like a librarian that puts the books together. Chromatin remodelers, chaperones, and polymers. So we're talking about chaperones, we're talking about manager proteins. Again, we're talking about signaling. Now, women with exceptionally youthful appearance and facial skin had an overall epidermal structure that was different, a barrier function in their facial epidermal samples, and dermal matrix production. Again, we're talking about gestalts. We're talking about interactions of different signals. And on the left, you see a non-healing phenotype, and on the right, you see a healing phenotype under the microscope. Now, this is the effects of what it looks like in a clinical case. This is a herpes zoster on an 86-year woman, and how signaling can transform that very, very quickly within 2 treatments and repair all the wounds. You also have very similar matrices that are affected in hair. So women with exceptionally youthful appearing facial skin, we also had a higher expression genes that are associated with the hair matrix production. And this is very new research from June et al. 2017. And this is some clinical studies of how signaling affects the hair. That's another one. And you can see as an improvement in the hair. Now, gene expression is regulated by genetic effects and by environment. The previous speaker, Dr. Yamada, spoke about that as well. So basically, there is an interaction, and again, this interaction creates another gestalt of signaling complexes within the organisms are interacting with the signaling complexes within the environment. They make a whole different complex that basically allow us to look young or to look old. So, is there a measurement for that? And yes, it is a measurement. There is the measurement is the quantitative trait loci and the quantitative trait loci analysis or locus analysis, and that's a statistical method that links phenotypic data with genotypic data and how those interact with each other. And quantitative trait locus is basically a region in the DNA, and the different types of quantitative trait loci create what we call the architecture of a trait. Now, for some reason, the skin seems to show the most age-related gene expression changes. As you can see on the diagram there, when you compare it to adipose tissue, it's 1,484 versus 185, so much many more genes expressed in the skin. And Glass et al. Found that the gene expression changes with age in skin, adipose tissue, blood, and the brain. And, but primarily it's the skin, and there are some genes that are basically the same, both in the skin and the adipose tissue, skin and blood, and skin and brain. They're the same kind of genes. And some of them are very tissue specific. Signaling pathways now are the key to this biological mechanism that create these intracellular and extracellular signals. Intracellular is within the cell, extracellular is outside the cell. And cell signaling plays a very key role in basically relaying extracellular signals and intracellular signals. And I'll show you a movie so you can see that. As you can see, the signaling mechanisms initiate these sequences from the outside of the membrane. These signals coming through inside the cell, and they affect the DNA. The gene is part of the DNA, and that gene information is used to synthesize proteins in the cytoplasm. And they have to process transcription and translation that are happening in the cytoplasm. And from the chromosome, you're going into the gene. From there, the DNA separates the RNA, travels into the cytoplasm, and this is the traveling, and gets into the ribosome, and that's where different amino acids are connected to make the proteins are basically the brains of our cells. The protein is a brain inside our cells. So basically what signal transduction is initiated by the binding of a ligand to a membrane receptor, like I showed you before. And then there's a downstream of signals that come inside the DNA, and they affect the DNA and the genetic, and that's where gene expression basically occurs. So the cells detect the signals from the outside, and then there is a translation and the transcription, and as the signals interact with DNA, we have the expression of a specific gene. So the coordinated activity of different signaling pathways within and between multiple cells is the basis for many biological processes, like in gene expression, of course, that basically affect human development at all stages. But what's interesting to see in the middle slide is that from 26,383,000 human genes, about 42% are unknown. We don't know what they're for. So there's still a lot to learn. And on the slide after that is different signaling pathways, the AMPK signaling, the calcium, and basically other pathways that affect the cells. Now, the response of biological systems to changing environmental conditions and aging factors is a dynamic process. It's exactly what we said before. It's a gestalt. It's more than the sum of its parts. Dynamic means many different things coincide together and create this other thing emerges that is totally than everything that a gestalt is like a personality. A gestalt is like beauty. It's not something you can explain or analyze. It's something that is more than the sum of its parts. And activation of these different signaling pathways is responsible for the cell proliferation, for cell death, for cell differentiation, cell metabolism. Basically, signaling determines and controls the entire anti-aging process. And a clear understanding of the activity and interaction between the signaling pathways can help to design rational diseases for treatment, and of course it has to do with the tissue regeneration and anti-aging. And on the left you see the cell count treatment in sensitive genes, and on the right you see the sensitive genes of a particular treatment. And here we have how basically we identify an important signaling pathway in the cell. We look at the gene expression, we look at the PPI, the protein-to-protein interactions, we look at general signaling pathway, the blueprint of the signaling pathway. Then we look at the map the cell, type-specific gene expression, and basically we combine gene expression and PPI. You're going to see this more in the next slide. Basically, you compute, it's a computerized expression of protein-to-protein interactions and gene expression combined together, and the interactive sum is the totality of PPIs that happens in a cell, and that is mathematically represented, and we look again at the gene expression. There is a lot of new methods that are far more thorough than ever before, the computerized methods like the HISP, a hybrid intelligent approach, and that combines protein-to-protein interactions and gene expression. And what you see here is just the signaling pathway for the cell, the cell of the wall of the cell, and how that controls the integrity of that cell wall. The blue is the beginning, the red is the ending of this signaling pathway. But as you increase in complexity, for example, this you have a signaling pathway in the human hematocytes. You see how much more complex this process becomes. And of course, the more you add, the more the complexity increases. Here is a schematic diagram of a molecular mechanism for osteoporosis. And then you have, of course, tropic hormones and signaling, you have intracellular crosstalks, you have attractive basins, and all of these are interacting in an endogenous network to create a gestalt that basically is responsible for either our osteoporosis happening or basically protecting against it. Our research started very, very long time in London University, and there was a paper that was published by Volping, and we were looking at basically RNA content in muscles, in skeletal muscle, not the heart or anything else, but the skeletal muscle. And basically there was a faster expression of the slow type genes, which are the genes that are related to the skeletal muscle. And there was, in Sunday Times, 1994, published an article on the content of the pacemaker. He started this signaling technology with the pacemaker. The pacemaker basically replaced signals of the pacemaker cells, and this technology replaces the signals of the motor nerve. It's more on the central nervous system, and since then we have not allowed to publish that much because everything is proprietary. This is the result of this technology after 25 years of research. This is just one treatment. This is two treatments, and this is just one treatment. And now you're asking, why am I showing you body pictures in a facial rejuvenation lecture or section? It's because the body, it controls the face. You cannot separate the face from the body. Don't cut the face off and you leave the body by itself. What you do in the body is going to affect what your face looks like, and your aging process will be affected both by the body and the face. And of course, a very important aspect of this is the gene expression of DNA repair, and younger women and older women with younger appearance have a greater ability to repair the DNA. In our research, what we do, we look at signals and how these signals can basically fuse with biological signals to either amplify them or to basically fill them in, like you fill in basically letters in a sequence that has lost its letters. And this is how we basically do it. It's computerized, obviously, and it's through signal cluster analysis, and you can see some of it here. And of course, the electronics have to do how you compose different waveforms to make complex waveforms, because a signal is not just one waveform. There's thousands of waveforms that are combined together. And there is another trick. It basically you can only put signals through the system in very, very low energies. So it's the exact opposite of lasers and radio frequency. This goes towards the complete other end of very, very minuscule energies. And in these energies, there's a mathematical proof, that's the book that was published in 2008, that it's mathematically proven that the ion channels of the cells open up. We don't know why, but they open up, and then you can insert signals or stem cell extracts for that matter. Now, there is another component that is extremely important. You know, everybody's looking at the watch, right? The clocks. The clocks control our lives. Why? Because we have clocks inside our bodies. Every little cell has a clock inside it. And basically, this is the Nobel Prize in Physiology and Medicine 2017 that was given because these investigators found that it's the molecular mechanisms, the cells, the clocks of the cells that control the entire circadian rhythm, that we cannot stay awake for 3 or 4 days. We have to go to sleep because our circadian rhythm demands that. And that is the gestalt from the different little clocks inside the cell that create this circadian rhythm. And clock genes determine several skin disorders, aging and disease. This is the clocks from leukemia. This is keloid scars. Keloid scars are also affected. So when we're talking about signaling, we're talking about basically taking into consideration these genetic clocks and incorporating them into the system, the computerized system for this technology. And this is after 20 minutes. This is stretch marks. Stretch marks are also, psoriasis is also as well an effect of circadian clocks. But also all of these skin disorders are the result of a dysregulation of the circadian rhythms. And you will ask, can we measure this system? Because it's like so minuscule and it's very difficult to obviously observe it under a microscope. And the answer is yes. We have a second virial coefficient, the SVC measurements. And when the SCV is basically we're looking at protein, if the proteins are aggregated, they're dysfunctional. They have to have a particular shape, and the shape of the protein determines the role that the protein plays. And if you have a negative SVC, then the protein aggregation is increased. That means you have dysfunctional proteins. If you have a positive SVC, the protein aggregation increases. Therefore, your proteins are functioning. That means your skin is going to look younger. And with this, I will conclude my lecture. Thank you very much. Any questions?